This is the process of transcription and translation, or in other words, how proteins are created from DNA. This is DNA, which has the nitrogenous bases as followed: red for adenine, blue for cytosine, green for guanine, and yellow for thymine. The first step in this process is to undo the helical shape of the DNA. This is called unzipping the DNA. Once the DNA is aligned, a section of the DNA splits into two parts in order to prepare to make mRNA. mRNA is copied from one side of the gene, a section of DNA, using the bases adenine, cytosine, guanine, and uracil. The order of the bases depends on the DNA sequence. If the DNA base is adenine, the mRNA is uracil. If it's cytosine, the mRNA is guanine. If it's guanine, the mRNA is cytosine, and if it's thymine, it's adenine. mRNA is single-stranded, unlike DNA. There is now a strand of mRNA in the nucleus. All the steps until this point have taken place in the nucleus of a cell. The mRNA travels to the ribosome to prepare to make proteins. 3 sections of RNA bases, called codons, code for an amino acid. In the ribosome, 2 codons, or 6 bases, code for 2 amino acids at a time. The codons also bind to anticodons, which determine the amino acid. The mRNA has a start codon and a stop codon. The ribosome starts coding a new protein when it finds the start codon, and it stops coding when it reaches the stop codon. These amino acids then form a polypeptide chain, which become proteins. In this model, it really only shows 2 amino acids, but in reality there are many amino acids in a polypeptide chain. The polypeptide chain makes a protein. Thank you for listening to this short video presentation.